Hello students, myself Srishti Singh, your biology mentor educator. Welcome to our Sankalp tutorial online platform. So let's start with a new lecture series for class 12 CBSE, that is sexual reproduction in flowering plants. So let's start. First of all, I will discuss about what is the structure related to flowers. So everyone knows that flower is a main site of reproduction in all plants because it is the main site where sexual reproduction take place. So in flowers there is a two holes. uh, One is essential hole or other is non-essential hole. So first we discuss about what is essential holes and what is non-essential hole. So essential holes. Essential holes are those which is essentially responsible for the reproduction in flowering plants because uh, if holes are absent in flowers, so there is no any sexual reproduction take place. While non-essential holes While non-essential holes are those which is not directly participate in sexual reproduction, but it's play a role in sexual reproduction, like attracting insects or birds to perform a pollination in flowering plants. So first we discuss what are the holes, what are the parts related to essential holes. So first one is that male reproductive parts. Male reproductive parts in flowers, and other is female reproductive parts. Because in sexual reproduction there is presence of sex organ is necessary, and in asexual reproduction there is no need of sex organ or a specialized organ to perform a reproduction. So what are the essential non-essentials hole in flowers? So non-essential holes are sepals, petals, and perianth. So these are the structure of flowers in which the first non-essential holes are petal, which is related to pollination in flowers because insects are attracted by these petals because they are colorful, and sepals are green in color, perform photosynthesis, and store a food material. After that, androecium and gynoeciums are major sexual organ in flowering plants because mm, androecium and gynoecium, sexual reproduction does not take place in flowers. So we discuss about what is androecium. So the unit of androecium is a stamen, which is further divided into three parts, that is anther, filaments, and this anther connected with filament by a tissue known as connective tissue. After we discuss about gynoecium. So gynoecium is divided. So unit of gynoecium is carpel, and this carpel is divided into three parts like stigma, style, and ovary. And this is a stalk of flower which is known as pedicel. So I think you will know about all the terms related to flower. So in these perianth, what is perianth? So when we combine petals and sepals, then it is known as perianth. After that, the male reproductive organ in flower, that is microsporangium. So when we cut the transverse section, when we cut the TS of anther, it looks like that is presence of MMC and tapetum. So in these microsporangium, there are two essential layers which is already present in this. That is, first one is epidermis and other one is hypodermis. In the hypodermis, the cell which contain dense cytoplasm and clear nucleus, further known as archisporial initial cells. So in anther, there is a two layer, that is epidermis and hypodermis. In hypodermis, some cells having dense cytoplasm and clear nucleus known as archisporial initial cells. These cells further goes for mitotic division and give rise to two types of cells, that is primary epidermal cells and primary sporogenous cells. These epidermal cells further give rise to three layers of microsporangium, and these primary sporogenous cells again divided and form microspore mother cells. So these three layers of microsporangium is known as endothecium, middle layer, and tapetum. And this primary sporogenous cell, which is further divided to form a microspore mother cells, again goes for meiotic division and form microspores. So the wall of microsporangium are there, and here is a four major wall, that is epidermis, endothecium, middle layer, and tapetum. The upper three layers, epidermis, endothecium, and middle layer, act as a protective layer in microsporangium. So, what are the protective layer of microsporangium? Everyone can answer. That is epidermis, endothecium, and middle layer. How can it form? So it form by mitotic division in archisporal initial cells, which is give rise to two types of cells, that is primary epidermal cells and primary sporogenous cells, and these primary epidermal cells further give rise to three layers of anther wall or microsporangium wall. So these are the three major layers which provide protection to microsporangium or microspore mother cells. After that, what is the function of tapetum? So tapetum provide developing tapetum provide nutrition to developing microspores. So protective layer of microsporangium is three layer, that is epidermis, endothecium, and tapetum. Sorry, epidermis, endothecium, and middle layer. And tapetum provides nutrition. What is the function of tapetum? So tapetum provides nutrition to developing microspores and or pollen grains. So after that, what are the function of microspore mother cells? So these microspore mother cells helps in the process of microsporogenesis. So what is the microsporogenesis? So microsporogenesis is a formation of microspores or pollen grains. So how can it forms? So microspores are formed by microspore mother cells, which is already formed by mitotic division in archisporal initial cells that form primary sporogenous cell, and these primary sporogenous cell further divide to form microspore mother cells. So these microspores further uh, divided by meiotic division. As you all know that in meiotic division, four cells are formed which contain half number of chromosome. So microspore mother cells, that is 2 and divided into 4 microspores, and ploidy is n. So these microspore mother cells divided by meiotic division and form 4 microspores And all these four microspores are attached to each other by callose. And callase enzyme break these spores and separate them as a pollen grains. So, in this nucleus division and cytoplasmic division, that is karyokinesis and cytokinesis, there are two types, that is simultaneous division and successive division. Okay, in simultaneous division, this nucleus is divided into two nucleus, that is karyokinesis 1, that is karyokinesis 1, and these were two nucleus further divided into four nucleus, that is karyokinesis 2. And after the completion of karyokinesis 1 and 2, cytoplasmic division take place. So this is known as cytokinesis. Again, listen. After the completion of karyokinesis 1 and 2, cytoplasmic division take place, that is cytokinesis, and tetrahedral pollen grains are formed. Okay, and other types is the successive division. In this division, one nucleus is divided into two nucleus, that is karyokinesis 1, that is karyokinesis 1 for first, and after the completion of karyokinesis 1, cytoplasmic division take place, that is cytokinesis 1, and after completion of cytokinesis first, again karyokinesis division take place, karyokinesis 2, and at last further cytokinesis division are there, that is cytokinesis 2. So in this process, karyokinesis followed by cytokinesis, then cytokinesis. So this type of division is known as successive division, and in these isobilateral pollen grains are formed. Okay, so these are the two major types of division, two major type of meiotic division which is take place in angiospermic plant. The major division which is take place in angiospermic plants are simultaneous division, in which karyokinesis, after completion of cytokinesis, karyokinesis take place, which give rise to tetrahedral pollen grains. So after formation of these four microspores, that is pollen grain, we we have discussed about the structure of pollen grains. So in structure of pollen grains, there is a two layers which is surrounded this pollen grain. So the pollen grains having a two layer. The first one is the exine and the second one is entine. So first layer in pollen grain is known as exine, which is made up of sporopollen, and this sporopollen is a highly resistant substance. This is highly resistant. So on this layer, there is no any effect of temperature, pH, light, UV treatment, okay, or any other physical factors. And the other inner layer is known as, this is exine is the outermost layer, and the other layer which is thin and innermost layer is known as intine, which is made up of pectocellulose. That is two component in this, pectin and cellulose. And this is a innermost layer. After that, in the pollen grain, there is two cells. That is, one is vegetative cells and other is generative cells. The vegetative cells are bigger in size and having irregular nucleus, while generative cells are smaller in size, just like floating in the cells. It seems like floating, uh, small, and having a spindle shaped nucleus. So this is a structure of pollen grains, and this structure of pollen grain is known as two-celled stage. In most of the angiospermic plant, the pollination take place as at a two-celled stage. That is 60% of angiospermic plant. 60% of angiospermic plant are there in which pollination are take place as and at a two-celled stage. When these vegetative nucleus, when this vegetative nucleus divided into two male gametes, this vegetative nucleus is divided. When this vegetative nucleus is divided into two male gametes, then this pollen grain is known as three-celled stage of pollen grain, and 40% of angiosperm, in 40% of angiosperm, the pollination take place. The pollination take place at three-celled stage. I think everyone is clear about that. So thank you so much. बढ़िया आप लोग. Okay. Bye.